Hello, future doctors. So my topic is all about glandular epithelium. Glands are composed of cells that are specialized in producing chemicals, and these chemicals can be secreted directly through ducts or directly into the body fluid, and most of the time that is the blood circulation. There are several ways on how we can classify glands. Number one, we can classify them based on the number of cells that we can find on that particular gland. That's why we have the terms unicellular and multicellular. If you only see one cell in the gland, you refer to it as unicellular. And there's only one example of unicellular gland in the body, and that is the goblet cell. So if you will have a matching type and you will be asked to identify whether the gland is unicellular or multicellular, just look for the goblet cells because that's the only unicellular gland. And then the rest of the question should be answered with multicellular. As mentioned, goblet cells are composed of a single cell. That's why it's described to be unicellular. What I want you to take note, the goblet cells are interspersed, inserted among the epithelial cells of respiratory tract and intestine. Respiratory tract is commonly lined by pseudostratified ciliated columnar epithelium. So if we're going to take a look at the lining epithelium of the respiratory tract, expect that inserted among the cells of the pseudostratified ciliated columnar epithelium are the goblet cells. These goblet cells specialized in producing mucus, which can have an important function for the respiratory tract organs and the intestines. We will elaborate these functions later on. If you want to confirm whether the cell you're looking at is a goblet cell, you can use the following stains. You have periodic acid shift and Alcian blue. Periodic acid shift will impart red color to the goblet cells, and the Alcian blue will impart blue color to the goblet cells. This is the lining epithelium of the respiratory tract. This is pseudostratified ciliated columnar. You can see the presence of cilia on the apical portions of the lining epithelia. Appreciate the light-colored cells inserted among the epithelial cells. These are now the individual goblet cells. So, one goblet cell is pointed by the blue arrow. Why do you think it's very important for the respiratory tract epithelium to have goblet cells inserted among the cells? Goblet cells specialized in producing mucus. Air that we inhale contains microbes, and for these microbes to be filtered out from the air so that once the air will reach the alveoli, it is already microbe-free, the microbes will be captured or trapped by the mucus. And the main purpose of the presence of cilia in there is to expel the mucus along with the trapped microbes out into the air every time we exhale. This is the picture of the trachea. It is one of the organs of the respiratory tract. Please take note that the trachea is supported by C-shaped tracheal cartilages. So expect that the trachea is supported by cartilage anteriorly and laterally. But since the cartilages are C-shaped, you're not supposed to find cartilage on the posterior end of the trachea. Let's have a cut section of the trachea and we will look at it through its lumen. So this is the cut section of the trachea. Can you appreciate the structure labeled as cartilage? It assumes a letter C shape. So that will provide support to the trachea anteriorly and laterally. But you don't see the C-shaped cartilage extending up to the posterior end of the trachea. Take a look at the area labeled as glands. On top of the glands is the lining epithelium of the trachea, and that's pseudostratified ciliated columnar. And as mentioned a while ago, the pseudostratified ciliated columnar of the respiratory tracts should have goblet cells. So what we will do is, we will view the lining epithelium of the trachea on a higher objective or magnification, and we'll try to prove whether the lining epithelium of the trachea really has goblet cells. And just like what I've told you, we'll try to view the trachea under high power objective or oil immersion objective. By the way, the blue arrow is pointing to the area where you can find the lining epithelium of the trachea. So we'll get a section of the trachea and view it under the high power or oil immersion. And this is how it will look like. So I want you to appreciate the cartilage support of the trachea. Then you have here the glands, because as you can see in the picture, the glands are immediately positioned beside the cartilage. Okay, so most likely the next labeled structure, okay, the one where you can see the sun symbol, okay, that is where you can find the lining epithelium. I want you to focus on the lining epithelium of the trachea. Please appreciate the structures pointed by the blue arrow. They are light staining, no? These are now the goblet cells inserted among the epithelial cells of the trachea. The lining epithelium, by the way, is pseudostratified, ciliated, columnar. Again, the purpose of the goblet cell is to produce mucus so that the mucus can trap microorganisms in the air. And the main purpose of the cilia is to create movement, ciliary movement, okay, that will propel now the mucus out of the body, okay? And just like what I've told you, goblet cells are present in the lining epithelium of the respiratory tract and the intestine. Okay, so this picture is an example of the lining epithelium of the intestine. So what do you notice? You have there a simple columnar epithelium because you only have one layer of cells. Okay, just look at their nuclei. No, the nuclei are almost at the same level. So that's actually how you should try to identify the number of layers of the epithelium. So looking at the cells there, their nuclei are almost at the same level. So it would only tell you that there's only one layer of epithelium. So you call it simple. And since the cells are tall, okay, so they look like columns, okay, so you will call them simple columnar. And what do you notice? There are whitish cells, okay, inserted among the simple columnar epithelium of the intestine. So those whitish cells in there are the goblet cells. As you can see, they are really unicellular. If the purpose of having goblet cells in the respiratory tract is to trap microorganisms in the air, okay, there's another purpose for the availability of goblet cells in the lining epithelium of the intestine, okay? And I want you to take note, the mucus that is produced by the goblet cell will actually provide lubrication so that as the food, okay, will move through the length of our small intestine and large intestine, the movement will not cause us to feel discomfort because the food, the digested material will just glide through the lumen of the intestine because there's mucus that will provide lubrication. So I want you to take a look at this picture. Those finger-like projections that you see in there are actually the villi of the small intestine, because if you can remember from your anatomy, the small intestine has villi. Okay, so those finger-like structures there are called villi. And I want you to take a look at the lining epithelium of the villi. Okay, so you can see that there's only one layer of cell because the nuclei are almost at the same level. Okay, and then the cells are tall, so therefore you will name the lining epithelium as simple columnar. And take a look at the colorless or whitish cells inserted among the columnar cells of the small intestine. Okay, so those are the goblet cells. Again, the purpose of mucus, okay, produced by the goblet cells in the lining epithelium of the intestine is to provide lubrication so that the peristalsis will happen without causing abdominal pain or discomfort. So just like what I've told you, there are special stains for goblet cells. Okay, we can either use periodic acid Schiff or Alcian blue. Periodic acid Schiff will particularly stain the mucin component of the mucus, which has carbohydrates, and periodic acid Schiff usually imparts red color. So I want you to take a look at the picture. Can you see finger-like projections? So those are the villi of the intestine. And you know very well that these villi are lined by simple columnar epithelium, and inserted among them are the unicellular goblet cells. So since periodic acid Schiff will impart red color, those red colored cells that you see in the picture, okay, the ones that are mixed along with the lining epithelium of the small intestine are the goblet cells. So let's have a closer look. So take a look at these cells. So they are composed of one layer of cells, and these cells are tall. So that will give you an idea that this is really from the intestine because it's simple columnar. And inserted among them are the red colored cells that are stained by periodic acid Schiff. So those are the goblet cells. Using Alcian blue, okay, from the name itself, the dye will impart blue color to the goblet cells. So this one will also prove to you that goblet cells are really unicellular and they can be incorporated in the lining epithelium of the respiratory tract and that of the intestine. So, based on number, glands can either be referred to as unicellular and multicellular, and the only unicellular gland in the body are the goblet cells. So, salivary gland, on the other hand, is an example of a multicellular gland. And I want you to take a look at this picture. We will try to sample the submandibular gland. This is how the submandibular gland will look like under the microscope. You can see multiple cells. So, therefore, all the glands in the body except for the goblet cells are multicellular. We can also classify glands based on where do they release their secretion or product. Okay? If they will release their secretion directly through a duct, so meaning they have ducts within their structure, so we will refer to them as exocrine gland. My Indian students would love this one to be pronounced as exocrine glands. Okay? If the gland is not provided with a duct, so therefore it is ductless, okay, you will refer to it as endocrine or endocrine. So I want you to take a look at the two photos. The gland on the right side of the screen, okay, has a duct, and you can see that the secretion is released through the duct. So therefore, this gland is called an exocrine gland. Okay, and I want you to take a look at the gland on the beside it. Okay, so you can see that gland doesn't have a duct. Instead, okay, it is surrounded by the red-colored blood vessels or capillaries. So that will tell you that endocrine glands release their secretion directly to the bloodstream. Okay, so let's proceed to the next slide. So both endocrine glands and exocrine glands are derived from the lining epithelium of the organs where they are located. So I want you to take a look at this lining epithelium. To form the glands, okay, there must be that proliferation of the cells, okay, that are derived from the lining epithelium. If the proliferating cells will still maintain their continuity with the lining epithelium where they are derived from, okay, then they will now form the exocrine glands. And the point of attachment to the lining epithelium is now called the duct. Okay, I want you to take a look at the red square there, and as you can see, the duct is labeled for you guys. Some glands, okay, will totally detach from the lining epithelium to which they are derived from, okay? But what do you notice? As these glands develop further, they are slowly, enveloped or surrounded by capillaries. So these glands now will eventually become the endocrine glands. So therefore, exocrine glands are glands with ducts, okay? While endocrine glands are ductless glands. Exocrine glands release their secretory substances or products into ducts, while endocrine glands release their secretory products directly to the bloodstream. If the product is released directly to the bloodstream or any other body fluid, it will now be referred to as a hormone. So let's try to identify these two glands. Okay, the one on the left has this structure that is lined by those white-colored cells, and as you can see, that structure is continuous with the lining epithelium. So that structure is actually the duct. So therefore, this gland is an exocrine gland. I want you to take a look at the gland on the right side of the screen. As you can see, the cells of the gland are surrounded by two red-colored structures. So those two red-colored structures are representing blood vessels. And as you can see, as indicated by the arrow, the cells are secreting their substances directly into the bloodstream. So therefore, that gland is an endocrine gland. So this is now the summary on how we form our exocrine glands and the endocrine glands. Okay, so they are both derived from the lining epithelium. Okay, and the lining epithelium would have cells that will replicate or proliferate, and as the cells proliferate, they will form a structure that will invaginate into the underlying connective tissue. Okay, if that growing structure will remain attached to the lining epithelium, you will call it as the exocrine gland. Okay, the proximal portion, okay, of that gland will become the duct. The distal portion now will become the secretory portion or secretory end piece. I will be showing you a picture later on. But if the developing structure will detach from the lining epithelium, then that one will mature to form the endocrine gland. So exocrine glands have two parts. The proximal part will eventually become the duct, okay? And that is the one that is continuous with the lining epithelium. The distal part, okay, will eventually mature to form the secretory portion. So that is where the substances and the chemicals of this gland are produced. Take a look at this picture. The yellow arrows are pointing to the distal portion of the gland, and this is the secretory portion of the gland, and the green arrows are pointing to the ducts. Now, on the succeeding slides, I will be teaching you how to identify ducts. So I will be showing you some pictures, and that is to train you how to identify the ducts among all the structures that you can find in multicellular glands. This is a picture of a multicellular gland, but focus on the areas pointed by the yellow arrows. Those are the ducts. Ducts can be lined by simple cuboidal or simple columnar epithelium as shown in the picture. Now, please familiarize yourselves with how the ducts appear under the microscope. Let's have this picture. Those structures pointed by the yellow arrows are the ducts. As you can see, they are lined by simple columnar epithelium. Let's have another example, and I want you to identify which among the structures that you can see in the multicellular gland is the duct. I want you to look for the ducts in this picture. I'll give you 5 seconds. This is one of the ducts. As you can see, it's lined by simple columnar. This is another duct. This is lined by simple cuboidal epithelium. Can you find one more duct? This is another duct, and it is lined by simple cuboidal epithelium. I'll leave this one an assignment for you guys. So I want you to look for the 4 ducts in the picture. You can send your answers to my Telegram so that I can verify whether you have identified them correctly or not. So let's have a review. Glands can be classified based on the number of cells that you can find in them. So that's why we have the terms unicellular for goblet cells and multicellular for the rest of the glands in the body. Okay. Then we can also classify glands based on whether they have ducts or not. That's why we have the terms exocrine glands and endocrine glands. Okay. This time we'll focus on the exocrine glands. So from this PowerPoint up to the last PowerPoint in this lecture, okay, we will be talking about glands with ducts or exocrine glands. So the third way on how to classify glands in the body is to base it on how do cells, or how do the cells in the gland release their secretion into the duct. Okay, so kanina we just want to know whether there's the presence of duct or not. This time we will try to identify how these glands, okay, will release the secretion into the duct. And we have 3 terms: the merocrine, the apocrine, and the holocrine. So let's try to differentiate these 3 types of glands. So we have here 3 types of glands, okay? And we will name them gland A, gland B, and gland C. So I want everybody to focus to gland B, okay? So as you can see in the picture, for the cells of the gland to release the secretion, they will release it to the duct through exocytosis. So I hope you can remember the term exocytosis, no? You have a vacuole inside which may contain the substance that this gland is supposed to release, okay? So that vacuole will fuse with the membrane in the process called exocytosis, and the substance now is released to the lumen, then into the duct, okay? So merocrine glands, okay, do not have to destroy a part of the cell for the product to be released, okay? The main reason why the product is released is exocytosis. So letter B is merocrine. Take note of that, ha? Let's go to letter C. Okay, what do you notice? As the cells, okay, will release the substance into the duct, they will lose a part of their apical portion. Okay, so as you can see in the picture, a portion of the apex of the cells are sloughed off or are removed from the cells, so that the products will be released into the duct. Okay, and since there's the loss of the apical portion of the cell, this gland, is referred to as apocrine gland. Okay, so letter B, merocrine, exocytosis. Letter C, a portion of the cell will be removed, and most of the time it's the apical portion of the cell. It's the letter C is now called as the apocrine gland. Letter A. Letter A releases its secretion by the destruction of the cells of the gland. So meaning the entire cell has to undergo lysis in order for the substance to be released. Okay, that's why you can see in the picture some of the cells are undergoing mitosis to replace the cells that has undergone lysis, just to release the chemical. Okay, and this one is called holocrine. Okay, so letter B is merocrine, exocytosis. Letter C is apocrine, apical portion of the cell is removed. But if the cell has to undergo total lysis just to release the substance into the duct, then you call it as holocrine, and that's letter A. So we will discuss these glands individually. Okay, so merocrine glands releases their secretion by means of exocytosis. Okay, you have to take note of that. So that's why, as you can see in the picture, there is no significant damage to the cells because the substance is just released through exocytosis. Okay, so by the way, the yellow colored cells there are representing the duct portion of the exocrine gland, while the red colored cells there are representing the secretory portion. I hope you can still remember these terms, no? Okay, so the three glands in the body, okay, that release their secretion through the merocrine or merocrine fashion are salivary glands, sweat glands, pancreas. Okay? For you to easily remember these glands, I want you to say, mero ako laway, laway, salivary gland. Mero ako pawis, sweat gland. Mero ako papa, pancreas. Okay? So, yan na. That's my way of helping you guys on how to easily memorize those glands that belong to the merocrine category or classification. So the next type of gland is the apocrine gland. So what can you remember? The apical portion of the cell is removed, okay, in order for the secretory product to be released. As you can see in the picture, pinch off apical portion of the cell, and that one will become part of the secretion. Okay, and there are two examples of apocrine glands in the body. Okay, mammary gland of the breast, okay, and ceruminous glands of the ear, the one that produces the cerumen or the ear wax. Okay, so every time you see a woman who is breastfeeding her child, okay, you sit by her side and say, do you know, you're not only giving nutrients to your baby, you're also sharing through your breast milk the apical portion of the cells of your mammary glands. Thank you. Okay, so every time you will also insert cotton buds into your ear. Okay, so as soon as you're removing the cerumen, you're also removing the apical portions of the cells of the ceruminous glands. So, yan na. We'll now move to the next type of the gland. So the next classification of glands based on the manner of secretion is the holocrine. So for the gland to be classified as holocrine gland, some of the cells of the gland will undergo total cell lysis just to release the secretory product. But what do you notice? The cells found at the base are undergoing mitosis to replace the cells that have undergone lysis. Holo, may nabasag. Holo, okay? And there are two types of holocrine glands, okay? So you have sebaceous glands of the skin, okay, and the meibomian glands of the eyelid. The sebaceous glands are actually the glands that drain directly into the hair follicle to release sebum. Okay, and sebum is actually there to keep the hair from drying. Okay, and sebum is rich in fatty acid. Fatty acid. So it can also have what? Antibacterial activity because the acid will kill the bacteria once they will try to enter the hair follicle and cause infection. The meibomian glands of the eyelid, on the other hand, are the special type of glands that you can find on the posterior side of the eyelid. So I want you to imagine they are found on the surface of the eyelid that is gliding through the cornea or the anterior part of the eye. Okay? So these meibomian glands function to produce what? Mucus, so that there would be what? Lubrication, so that as the eyelid will glide through your cornea or eye, okay, you will not feel any discomfort or pain. Okay, so if you're blinking your eye in front of your crush and he or she will ask you, what are you doing? Instead of saying na nagpapakyut ako, you will just say, I'm just using my meibomian glands. So I want you all to take a look at this picture. The salivary gland, mero ako laway, okay, is an example of a merocrine gland. So as you can see, it releases its secretory product via exocytosis, okay. Mammary gland is an example of an apocrine gland, okay. What do you notice? Just follow the white arrow. The apical portion of the cell has to be removed, okay, in order for the secretory product to be released, okay. And then labeled in the photo, the sebaceous gland, okay, what do you notice? This one is an example of an holocrine gland. So if you're going to appreciate the picture, okay, you can see that the cells on top are undergoing lysis while the cells at the base are undergoing cell replication. So we have already classified glands based on the number of cells that you can find in them. We have also classified them based on whether they have ducts or not. Then, given that they have ducts, we have also classified them on how do they release their products into the ducts. This time, okay, we are not concerned with how do they release their products. We are concerned with what are they releasing. Okay, so meaning, regardless of whether it's exocytosis ba or cell lysis or removal of the apical portion of the cell, the next classification would deal much on what is the content of the secretion. Okay? So, we can classify them based on the type of their secretion, and we can classify them as two, serous or mucus. Okay? And I want you all to listen very carefully. If the cells, okay, are dark stained, that's your clue that the cells are serous cells. And these serous cells are producing enzymes, okay, or proteins. So, serous, dark, protein, enzyme. If the cells of the gland are light stained, okay, that's your clue that those cells are mucous cells. And from the name of the cells, they are producing what? Mucus. Okay? And I want you to take a look at the cells in the picture. One is dark stained and it's labeled serous. Take note of that, ha? One is labeled mucus. Okay? And I want you to appreciate the nuclei of the serous cells are situated in the center and it's spherical. And they are spherical, I mean. While the nuclei of the cells of the mucous cells, okay, the nuclei of the mucous cells are found at the base and they are flattened. So aside from the staining characteristic, aside from whether identifying whether they are dark stain or light stain, you can also look at the nuclei. So I will give you examples of pictures of specimens that have serous cells and mucous cells. Okay. So I want you to take a look at this picture. Can you see a duct? Okay. So I know you can already identify a duct, because I have already trained you on the previous slides. Okay. You look at the cells, okay, surrounding that duct. Okay. I don't mind those yung white colored cells. Those are just adipose tissue. You look at the cells surrounding the duct. Okay. Can you appreciate that they are dark stained? Okay. And they have spherical nuclei, typical of a serous cell. Diba? Serous cells are dark stained. Okay. They have round spherical nuclei almost found at the center of the cell. So therefore, this gland, you are pretty sure that this gland is enzyme producing or protein producing. Let's move to the other picture. Okay. Can you identify ducts in the picture? I think I can see two big ducts. Okay. So, you look at the cells surrounding the ducts. Okay. Can you appreciate that they are all light stained? Okay. And then look at the nuclei of those cells. They are situated at the base and they are flattened. So, therefore, you're looking at mucous cells. So, again, dark stained cells are serous. They produce enzymes and proteins. White colored cells, okay, or light stained cells, I mean, the better term is light stained cells, okay. They are referred to as what? Mucous cells, and they produce what? Mucus. So let's try to apply what you have learned from the last two PPT slides on this particular photo. The yellow arrow is pointing to a duct. Okay, that's very clear na. Next, I want you to take a look at the green arrow. Okay, as you can see, it's pointing to a dark staining gland. Okay, so most likely that will tell you that that gland is a serous type of a gland and it's capable of producing what? Enzyme and protein. So take a look at the nucleus or the nuclei of the cells of that gland. They are spherical and somehow they are found almost at the center of the cells. You look at the red arrow. Okay, it's pointing to a light staining gland. So most likely that will give an idea it's a mucus gland. And I want you to observe the nuclei of the cells of that gland. The nuclei are found at the base, okay, and they are flattened. So you will now have an idea that this gland can produce both an enzyme and a mucus. Okay, so let's apply what you have learned from the very beginning. Classify this gland based on the number of cells. Most likely you will say what? Multicellular, because you can see hundreds or even thousands of cells in there. I think hundreds lang. Next, exocrine or endocrine. Since the yellow arrow is pointing to a duct, you know that this is a what? Exocrine gland. Very good. And then I want you to determine, okay, the type of secretions that it can produce. Since you see serous cells, you also see mucous cells, you know that this gland is capable of producing both mucus and enzymes or proteins. So in this photo, you have here the yellow arrow pointing to ducts, okay? And then take a look at the red arrow. The red arrow is pointing to a light stained gland, so most likely that is a mucus gland. And look at the nuclei, they are basal and they are flattened. And then the green arrow is pointing to a serous gland. You look at the staining reaction, it's dark stain. And then look at the nuclei, okay? They are round in shape and they are almost found at the center of the cell. So there are two shapes for glands. Okay, we have the terms tubular and we have the term acinar. Some books would refer to the term acinar as alveolar. Okay, take a look at the blue arrow. The blue arrow is pointing at a club-shaped gland. Okay, so if the gland has a club shape, then you refer to that as acinar or alveolar. Okay, so that's the shape of the gland. If you're going to make a thin section of that gland and view it under the microscope, you will see that the cells of that particular acinar gland, okay, are dark stained. So therefore, you can assume that the acinar glands are serous glands because their cells are dark stained. So they are capable of producing proteins and enzymes. So therefore, we can conclude at this point, okay, that if you see a dark staining gland under the microscope, you can always imagine that the shape of the gland, okay, before it was cut into thin section, okay, is acinar. You look at the yellow arrow. The yellow arrow is pointing to a gland that has the shape like that of a test tube. So that's why it's called tubular gland. And do you know that majority, okay, no, all of the cells in the tubular glands are light stained. So meaning they are what? Mucous cells, and they are producing mucus. So looking at this picture, okay, since this gland has both the acinar slash serous, okay, gland, and it also has tubular slash mucous gland, so therefore you can assume that this gland is a mixed type. So it has what? Serous glands, it has mucous glands. The last but not the least, okay, of classifying gland is based on their morphology. Okay, so let's start with the terms simple and compound. So looking at the pictures, you know that these glands are exocrine glands because they have ducts, and the ducts are colored blue. Okay, if the duct of the gland is unbranched, so meaning it's not forming branches, so you call the gland as simple. So the first row of glands, okay, as you can see, their ducts, colored blue, are not branching. So therefore, you will use the term simple to refer to them. I want you to take a look at the second row of glands. Can you appreciate that the ducts are branching? They are creating smaller branches. So that's the time you will use the term compound. Okay? Then, we will have the terms acinar slash alveolar and tubular. And we have already explained this one a while ago. So, I want you to take a look at the first gland. Okay? As you can see, the gland is shaped like a test tube. So, most likely that gland is tubular in shape. Okay? And then, I want you to take a look at the fourth gland. First row ha, fourth gland. Start from the left to the right. As you can see, the secretory portion of the gland has a club shape. Okay, so that is most likely an acinar or alveolar gland. So, in determining the morphology of the gland, we need to combine the description that we have for the duct and the shape of the secretory portion. So let's try to apply that one on the next PowerPoint slide. So all of the glands that are shown in these pictures, okay, have unbranching ducts. Okay, the ducts are represented by the yellow colored structure. Okay, so since all of their ducts are not branching, all of them are referred to as simple glands. Okay, I want you to take a look at the first gland on the left side of the photo. Okay, what do you notice? The secretory portion is tubular in shape. Okay, so since you know that the duct is not branching and the shape of the gland is tubular, you will just combine the two terms. So that first gland is simple tubular. Okay, next, let's move to the next gland. Okay, as you can see, the duct is still not branching, so we'll use the term simple. But what do you notice about the gland? The secretory portion, I mean. The secretory portion of the gland is branching. So it's very important that you should refer to it as branch tubular. Okay? So, the duct is described as simple. The secretory portion or the gland is described to be branch tubular. So, you just have to combine the two. Simple, branch, tubular. I hope naintindihan ha? Next. Let's move to the third gland. As you can see, the duct again is not branching. You use the term simple. But when you look at the secretory portion, it's club shaped. So most likely that's an acinar or alveolar. So just combine the two terms, simple acinar or simple alveolar. Okay? Then move to the last photo, the one to the right side of the screen. The duct is not branching. You use the term simple. Okay, and look at the gland. The gland is actually, the secretory portion is actually branching. So you will describe it as what? Branch acinar. So put them together, you have simple branch acinar. So let's move to the next slide. So as you can see in this group of glands, the ductal portion of the gland is branching. So I want you to take a look at the yellow colored portions of the glands. Can you see, can you appreciate that they are branching? So instead of using the term simple, you will use the term compound this time. Okay? So look at the first gland. As you can see, the duct is branching, and at the terminal ends of the duct, okay, you have there the secretory portions, and they are tubular in shape. So you will just simply refer to it as compound tubular. Second gland, duct is branching, use the term compound. Look at the shape of the secretory portion. They are club shaped. So you will use the term acinar or alveolar. So you just can simply refer to this as compound alveolar or compound acinar. Okay? The third photo, okay, the third gland, I mean, the one on the right side of the screen, has tubular shaped glands and club shaped glands. So this is actually a combination of both tubular glands and acinar glands. So that's why you refer to it as compound tubulo-acinar or compound tubulo-alveolar. Okay? So let's try to discuss the simple group of glands. Let's focus on the gland on the left side of the screen. You have there a non-branching duct, so you will use the term simple. And then take a look at the secretory portion or the glandular portion. Can you appreciate that it is tubular in shape? So this gland is most likely a simple tubular gland. The gland beside it as a non-branching duct, but what is branching is the secretory or glandular portion. The glandular portion is tubular in shape. So you will refer to this gland as simple branch tubular. Simple because the duct is not branching. Branch tubular because the glands are branching and they are tubular in shape. Take a look at the gland in the center. It is simple tubular, but the glandular portion or the secretory portion is coiled. That's why this one is referred to as simple coiled tubular. But this is basically almost similar to simple tubular. It's just that the secretory portion is coiled. The gland beside it has non-branching duct, so you will use the term simple, but the secretory portion is club-shaped. So you will use the term acinar or alveolar. So this gland is simple, acinar or alveolar. The gland beside it, the one on the rightmost side of the screen, has a non-branching duct, but the glandular or secretory portion is branching, and the secretory portion is clubbed shaped. So you will use the term simple because the duct is not branching, simple branch alveolar or simple branch acinar. In naming compound glands, we no longer use the term branch. The term branch is only used, okay, if the secretory portion is branching in simple types of gland. In a compound type of gland, we do not anymore use the term branch. Okay, so it's enough to use the term compound, and it would mean that the ducts are branching and as well as the glands. Okay, so kasi nga compound, when you say compound, two. So meaning dalawa nagbabranch. So that would refer to both, okay, duct and gland. Because if you're going to say compound, branch, acinar, redundant na siya. Okay? So compound, dalawa, duct and gland are branching. So yan yung katunang pasabot ato. So let's try to take a look at the first photo. Okay? The duct, the violet portion is branching. And look at the glands, they are also branching. Two of them are branching. So use the term compound. And look at the shape of the gland, they are tubular. So use the term compound, tubular. Okay? So the second one is most likely compound, acinar. Okay? And then the last one, take a look at it. There are portions, okay, with branching acinar glands. There are portions with branching tubular glands. But since the duct is branching, so both the gland and the duct are branching, use the term compound. So just combine compound, tubulo, alveolar, or compound, tubulo, acinar. So what we will do with the succeeding slides is to identify where can we find examples of these glands in our body. Simple tubular is actually found, okay, in the colon or the large intestine. So I want you to take a look at one gland shown in the photo. As you can see, the blue arrow is pointing to its duct, okay, ductal opening. So as you can see, it's not branch, so use the term simple. You look at the red arrow is pointing to the secretory portion of the cell, okay. Look at the shape, it's shaped like a test tube. So you will say simple tubular. Colon ha, large intestine. So this is again a picture of the cross section of the large intestine or colon. The blue arrow is pointing to the duct of the gland. So as you can see, it's not branching. You will use the term simple. Red arrow is showing you that the gland has a tubular shape. Okay, so therefore, colon has simple tubular gland. Can you appreciate the goblet cells that are interspersed within the simple columnar epithelium of the colon? Okay. So they, 'di ba? They function for what? Lubrication. So let's try to check how the colon would look like after you will apply Alshan blue stain, which is specific to goblet cells, right? So move to the next slide. So this photo is showing you the simple tubular glands of the colon, okay? And since you have learned from the previous slide that the colon has goblet cells in it and we can apply Alshan blue, okay? So after staining the colon with Alshan blue, you will now start to see the blue colored goblet cells in the lining epithelium of the colon. So this one is an actual photo of our colon slide specimen in histology laboratory in the MSF. So as you can see, the red arrow is pointing to a simple tubular gland. Okay, so simple tubular gland is present in the colon. But there is that another area in the body where you can find a simple tubular gland. But this time, it's shorter than that of the colon. So I am talking about the small intestine. Small intestine has villi. So the finger-like projections that you can see in the picture are the villi. So every time the villi will project upwards, okay, as it goes downwards, can you appreciate that there is that small invagination that it can create before it will again project upwards, okay? That small invagination is actually called the crypt of Lieberkühn, Okay? And take a look at the shape of the crypt of the liver con. It's actually what? Simple tubular. So that is again another example of what? A simple tubular gland in the body. So this is how the small intestine would look like under the microscope. So the blue arrow is pointing at the tip of one of the villi of the small intestine. And what do you notice? As the villi will project downwards, okay, it will actually create a small invagination, the one pointed by the red arrow. So that is now your crypt of Leverkusen. That's how you're supposed to pronounce it. Leverkusen. Okay? I just checked Google kasi. So, Leverkusen. Okay? So, take a look at the shape of the Leverkusen. It's actually simple tubular. So, we have two types of simple tubular glands. The colon and the crypts of ikaw ha, Leverkusen, pud ka ha, of the small intestine. So the next gland that we will discuss is the sweat gland. So I want you to take a look at the labeled sweat gland in the photo. You look at the duct first of the sweat gland. As you can see, the duct is not branching, right? So therefore, you will refer to this gland as a simple type of a gland. You look at the secretory portion, okay? It's actually shaped like a tube. So therefore, this is a tubular gland. But what I want you to appreciate in the photo is that as the sweat gland will invaginate deep to the dermis, okay, the terminal portion of the duct and majority of the secretory portion will actually coil. Okay, so therefore, you will describe the morphology of the sweat gland as simple coiled tubular. Simple because the duct is not branching, coiled tubular because the secretory portion of the gland is coiling. It's forming a ball of spaghetti. So again, sweat glands are simple coiled tubular glands. This is how the sweat glands will appear under the microscope. And most of you would wonder, huh? I'm supposed to expect spaghetti looking gland under the microscope, but how come I only see this particular microscopic detail? So I will try to explain to you how those sweat glands look like that one in the picture when in fact they are coiled tubular type of a gland. So this is the photo of the sweat gland. So as you can see, the yellow colored portion there is representing the duct of the gland. Then the red colored part is representing the secretory portion of the sweat gland. Okay. Then I hope you can still remember from our topic in the first lecture in histology that in order for us to observe the microscopic details of the different tissues or organs in the body, we must always prepare thin cut sections. And then what we will do, we will place these thin cut sections on top of the slide, stain them, then view them under the microscope. So let's say, for example, we're about to make a thin cut section of this skin specimen where we can find the sweat gland. So what will happen? As we make the thin cut section using the, let's say, microtome, okay, we will be cutting through the duct. So therefore, the duct will be seen under the microscope, okay? And then, as we go deeper, we will also be cutting through the proximal portion of the secretory part of the gland. But what do you notice? The portion next to the proximal part of the gland will actually curve forward. So therefore, that curved portion of the sweat gland will not be included in our thin section because I want you to imagine that if to prepare thin section, we must always make a straight cut. Okay? But so, therefore, this portion will not be seen under the microscope. Okay? Then what will happen? As that portion of the secretory part of the gland will curve again back to the area where we are making the thin cut section. So that particular portion of the secretory part of the gland will again appear under the microscope. So that's how you're supposed to imagine how sweat glands will appear under the microscope. So let's try to apply that particular concept on this photo. So let's say we're about to cut prepare a thin cut section. So as we are doing the cutting or sectioning, we will be cutting through this portion of the gland, okay? And imagine the portion next to it will actually curve forward. So therefore, it will not be included in the cut section. But as it curves back to the area where we are cutting, okay, that portion now will again be present in the specimen that we are preparing. So the same story goes with the other structures that you can see under the microscope. So just try to imagine na lang that all of these circular structures that you see are parts of the sweat glands, and they are continuous with each other because the gland is coiling. So on the next PPT, I actually prepared a sample video of how are you supposed to imagine how sweat glands are cut using a thin cut section, okay, in histology. So just watch the video and just try to understand what am I trying to tell you, because actually I was not talking while I was doing or recording the video. So I'll try to provide na lang an explanation with what I'm trying to say, no, or to show you in the video. So as you can see, I'm demonstrating here how a simple cold tubular gland will look like. Okay, and then I'm showing you that we're about to make a straight thin cut section. So what will happen? As we cut the individual parts of the gland, they would appear as circular structures. Okay, so you have already made like 4 circular structures, and if you see them under the microscope, just always imagine that they are connected to each other because the gland is coiling. Mura makugtangaan ni sa video eh. Sige na lang. So therefore, this is how sweat glands would appear under the microscope. So those circular structures there are actually linked to each other because they are forming a continuous coiled tubular gland. So this is just another picture of a sweat gland. So this is how a simple coiled tubular gland would appear under the microscope. So this is the best photo of the sweat gland that I can find in the internet. So this is the skin. As you can see, it's lined by stratified squamous keratinized epithelium. And the structure pointed by the blue arrow is the duct of the gland. So since it's not branching, you will refer to it as simple. And then appreciate the yellow arrow. So that's the secretory portion of the gland. So as you can see, it really resembles the appearance of a simple cold tubular gland under the microscope. So we will now move to the next type of a gland, and that's simple branch acinar. And the best example I can give you is the sebaceous gland of the skin. Okay, so I want you to appreciate the photo. You have there a hair. Okay, so that hair is embedded in the hair follicle. I want you to take a look at the gland that is draining into the hair follicle. The red arrow is pointing to its duct. Okay, you look at the duct. The duct is not branching, so therefore this gland is referred to as a simple type. Okay, you look at the secretory portion that is pointed by the blue arrow. Can you appreciate that the secretory portion is club shaped, and when it appears to be club shaped, then you call it as acinar. But look at the secretory portion of the gland. The acinar glands are actually branching, so you will refer to the duct as simple. You will refer to the secretory portion as branch acinar. So therefore this is a simple branch acinar gland. So let's have another example of a picture of a sweat gland. So as you can see here in the photo, you have the hair, okay, and then the hair is embedded in the hair follicle. Attached to the hair follicle, the one labeled as seb, okay, it's actually your sebaceous gland. So as you can see, it really resembles the sample photo that we have in the previous slide. So it's really a simple branch acinar gland. Okay, and since this is the skin, you should expect to find sweat glands. You take a look at the portion in the picture that is labeled as sweat. So the arrows are actually pointing to the sweat glands, and you look at their appearance. So they really appear to be what? Simple cold tubular glands. So this is the skin. As you can see, you have the hair in there. The hair is actually arising from the hair follicle. The yellow arrow is pointing to the duct of the sebaceous gland. Okay, as you can see, it's not branching. So this is really a simple type of a gland. You look at the white arrow. The white arrow is pointing to the acinar gland of the sebaceous gland. As you can see, there are 3 acinar glands in there. So this gland is really a branch acinar type of a gland. So sebaceous gland is a simple branch acinar gland. Look at the blue arrow. What do you notice? They look like what? Sweat glands, right? Because they are what? Simple, cold, tubular. Anyways, this is the skin. So expect that you can see sweat glands, hair follicles, and sebaceous glands in it. So this is another picture of the sebaceous gland. So I hope you can already memorize how it appears under the microscope, ha? Simple, branch, acinar gland. So we have already discussed simple tubular, simple coiled tubular, and simple branch acinar. Simple tubular colon, crypts of liver con, and then you have the simple coiled tubular represented by the sweat glands, and then you have the simple branch acinar, best exemplified by sebaceous glands. So let's now go with simple branch tubular. As you can see, these glands are provided with non-branching duct, okay, and you look at the shape of the secretory portions, they look like test tubes, right? So they are really tubular, but you can see several tubular structures in there. So therefore this gland has a branch tubular secretory portion. So this one is a simple branch tubular gland. So the good example of simple branch tubular gland in our body is the gastric gland, the gland of the stomach. So the blue arrow is pointing to the ductal portion of the gastric gland. Okay, so as you can see, it's not branching, so you will use the term simple. And then the yellow arrow is pointing to the secretory portion. As you can see, the secretory portion is tubular. But then again, you can clearly see that the tubular portion of the gland is branching. So therefore, gastric glands are simple branch tubular glands. So this is a cut section of the stomach. You look at the red arrow. The red arrow is pointing to the duct of the gastric glands, okay? And this duct will lead you to two tubular glands. So therefore, the tubular portion of the gastric glands are really branching. So this is simple branch tubular. So this is the picture of the histology of the gastric gland. The green arrow is pointing to the duct of the gland. The blue arrows are pointing to the secretory portion of the gland. So what do you notice? The secretory portion is tubular in shape. But for that particular duct that is labeled by the green arrow, you have two tubular glands attached to it. So therefore, the secretory portion is branch. So how will you classify this gland? Simple branch tubular. Again, I want you to appreciate where's the duct and you look at the branching tubular portion of this gland. So let's have a review first of the first few glands that we have discussed already before we'll move to the compound group of glands. So the first photo is showing you a simple tubular gland. Simple because the duct is not branching. Tubular because the secretory portion looks like that of the test tube. Okay. Example for simple tubular, the colon, the crypt of Lieberkühn. Okay. The second photo is a simple coiled tubular gland, best exemplified by our sweat glands. Okay. The third one has a non-branching duct, so therefore you will use the term simple. And then when you try to examine the secretory portion, you have tubular shaped glands, and you have more than one tubular shaped gland in there, so therefore you will call it as a branch tubular gland. So it's a simple branch tubular gland exemplified by the gastric glands. Okay, and the fourth one is the simple acinar. And take note, we do not have a simple acinar gland in our body because this simple acinar is only present when we were still an embryo. So I hope you will remember that one, ha. The last one, okay, is a simple type of gland because the duct is not branching. It's called acinar because you look at the shape of the glands, they look like club shapes. They have this club shape. They are described to be club shape. Yung pala yung better na statement. Okay? But you see more than one acinar gland. So this is a branch acinar gland. So simple branch acinar. Good example, sebaceous gland. We will now move to the compound type of gland. Papa, duo with mama, papa, mama, duo, sub-sub. Okay? So that's making it easy for you to memorize the glands and their morphology. So we will now discuss compound acinar glands. Remember, acinar glands have dark staining cells. So therefore, if they are dark staining, you will call the gland or describe the gland to be a serous type of a gland. And you know that it will produce what? enzymes and proteins. Remember, when you use the term compound, okay, it means lang that the duct and the gland are both branching. So how do compound acinar glands appear under the microscope? So let's move to the next slide. So I want you to take a look at this photo. The blue and the red arrows are pointing to ducts. Can you appreciate the ducts in the picture? Okay, so the presence of two ducts in the picture would only tell you that the duct is branching. Because if the duct is not branching, you're only supposed to find one particular duct, and that duct is surrounded by the secretory portion of the gland. But in this picture, you can see two ducts, so meaning the duct is branching. So you will have in mind that this gland is a compound type of gland. Okay, you look at the cells surrounding those ducts. Can you appreciate that they are all dark stained? So therefore, these cells are serous cells, and they are capable of producing proteins and enzymes. So if you will be asked what's the shape of the gland, you always have to associate serous cells to acinar shape. So two ducts, compound, dark stained glands, serous equal acinar. So just combine the two terms, compound, acinar. So this could be either the parotid or the pancreas. So let's have another example, ha. So I want you to take a look at this photo. You can see multiple ducts, right? So you will have in mind that this gland is a compound type of a gland. Okay. And you look at the cells or the glands surrounding those ducts. They are all dark stained. So therefore, these cells are what? Serous type. So if you will be asked what is the shape of the gland, what will be your answer? Acinar or alveolar. So therefore, maraming ducts, multiple ducts, compound, dark stained cells, serous, shape is acinar. So you'll just combine the two terms, compound, acinar. Example includes what? Parotid and pancreas. So that's why this particular picture is entitled Slide 87 Parotid Gland. So let's have another example so that you will really learn from this discussion. Can you appreciate multiple ducts in the picture? Yes. So therefore, this is a compound type of a gland. You look at the cells surrounding it, all of them are dark stained. So this is what type of a gland. So this is what? Compound acinar. So if I will use this picture in the quiz and ask you what is the product of this gland, what will you answer? Protein or enzyme. If I will ask you how will you classify it based on the number, you will answer what? Multicellular. Is it an endocrine or an exocrine? Since you can see ducts in the picture, you will classify this one as what? Exocrine. So the next type of compound gland that we will be discussing is the compound tubuloacinar. So from the name of the gland, no, tubuloacinar. So expect to see tubular glands which are composed of light staining mucous cells. Okay, then you have acinar shaped glands which are composed of dark staining serous cells. So expect to see the combination of dark stain and light stain glands or cells in this particular type of gland. Since you use the term compound, expect to see multiple ducts. So let's try to discuss an example of a photo. So try to take a look at this picture. Okay, the yellow arrows are pointing to ducts. So that will give you an idea. So this is a compound type of a gland. Okay, you take a look at the red arrow. Can you appreciate that the red arrow is pointing to a light staining gland? You look at the nuclei of the cells. They are at the base and they are flattened. So that will tell you that these cells are mucous cells. So most likely the red arrow is pointing to a tubular gland. Okay? So you already have come up with the term compound because there are two ducts, and tubular because there's that light staining gland in the specimen. Okay? You look at the blue arrow. The blue arrow is pointing to dark staining cells, and they have spherical nuclei almost situated in in the center of the cells. So these are serous cells, and serous cells have what shape? Serous glands have what shape? Acinar. So this gland is a combination of a tubular gland and an acinar gland. So since there are two ducts, use the term compound and then combine tubular and acinar. So therefore, this gland is a compound tubuloacinar or compound tubuloalveolar gland. Another example, next slide. So the yellow arrows are pointing to ducts. Okay, you can see multiple ducts in there. So automatically, you have an idea that this is a compound type of a gland. Okay, you look at the blue arrow. The blue arrow is pointing to dark staining cells. So those are serous cells. So therefore, there is that acinar gland. Okay. Then you look at the red arrow. The red arrow is pointing to light staining cells. So those are mucous cells. So therefore, you can conclude that this gland is a compound acinar. What are the examples again? Mammary, yes, mama sub, right? Mammary, submandibular, okay, and sublingual. I want to make this clear na as early as now. Some books would say mammary is compound acinar, but majority of the readings that I had would say that the mammary gland is compound tubuloacinar. Okay, because one of the photos that I use in this PowerPoint presentation labeled the mammary gland as compound acinar. Okay, but I believe, because it's mentioned in most books, that mammary glands are compound tubuloacinar glands. Okay, so let's review. Compound acinar, pancreas, parotid, papa. Okay, compound tubuloacinar, mama, sub. Mammary, submandibular, sublingual. So let's now move to the last type of a gland under the compound types of gland, and that's the compound tubular gland, the duodenal glands. So expect that all the glands that you see in there, okay, are light stained. So this is an example of the picture of the Brunner's gland or the duodenal gland. So do not mind the word Brunner's gland. We'll have that in our digestive system. So it's enough that you know that term duodenal glands. You look at the glands. All of them are basically light stained. So therefore, you have what? Multiple tubular glands in there. So this is compound tubular gland. Example, duodenal glands. This is again another example of a compound tubular gland. As you can see, all the glands in there are light stained. So I came up with a practice quiz. So this will help you assess the extent of your understanding of the topic. So what I want you to do is identify the morphology of the gland and give example. So how will you know if you're correct? You can chat me, okay, and ask me if your answers are correct. I won't mind if all the 600 BSMLS students will chat me, okay, because I really want to help you guys understand my lecture. Okay, so please be reminded that the quiz next Saturday will include Dr. Solano's topic on nucleus and glandular epithelium. Good luck, everyone. So I'll give you 10 seconds to give me the morphology of this gland, okay, and also give me an example. So ready? Time starts now, I mean. Okay, the correct answer. This gland is a compound tubuloacinar. Why? Because I can see 3 ducts in there. Okay, you have to label them. You have to identify them. I mean, there are glands that are dark stained. Okay, so therefore those are acinar glands. There are also glands that are light stained. So those are tubular glands. So this gland has both tubular glands and acinar glands. So this is a compound tubuloacinar. Next, can you identify the morphology of this gland? This is what? Simple tubular. Examples? Colon. You have the crypts of Libercon. How about the gland shown in this picture? This is simple branched acinar. Very good. How about this one? Can you appreciate that there are several ducts that you can see in the picture? So that will only give an idea that this is a compound type of gland. Take a look at the cells or the glands surrounding the ducts, and they are all dark stained. So This one is compound acinar gland. Okay. And that ends our lecture on glandular epithelium. Thank you very much for listening. Okay, so when you say compound, you expect that the ducts and the glands are branching. So therefore, you should never use the term branch for a compound group of glands because the compound word already means that both the duct and the glands are branching. So let's take a look at the photo on the upper part of the screen. Okay, you have there compound gland because the ducts are branching. When you take a look at the what you call this, the shape of the glands, they are tubular, right? So you'll just simply call it as compound tubular gland. Okay, then you look at the two glands, okay, found on the lower part of the screen. Okay, you have their compound acinar on the left side of the screen because the ducts are branching, use the term compound, and the shape of the glands are club shape, so therefore you refer to them as acinar. Okay, so compound acinar. The one on the right side has both tubular shaped glands and club shaped glands. So that's a combination of what? Tubular and acinar glands. So you will call it as compound tubulo-acinar. Compound because again, the ducts are branching. So let's discuss the examples, ha. Okay, sige. For compound tubular, you have duodenal glands. We also call them as Brunner's gland. Next, for compound acinar or compound alveolar, you have pancreas and parotid. For the compound tubulo-acinar or compound tubulo-alveolar, you have mammary gland, you have submandibular and sublingual. And for you to easily remember, no, the names of these glands, I want you to say Papa, pancreas, parotid. Papa, duo with mama, merong masubsob. Okay? So, let's apply that one on the next PowerPoint slide.